Flea-Borne Rickettsioses, a group of infectious diseases caused by various species of bacteria belonging to the genus Rickettsia, pose a significant public health concern worldwide. These pathogens are primarily transmitted to humans through the bites of infected fleas, which act as vectors for the bacteria. Among the most notable members of this group are Rickettsia typhi, the causative agent of murine typhus, and Rickettsia felis, associated with flea-borne spotted fever. The complex life cycle of these bacteria involves both arthropod vectors, such as fleas, and mammalian hosts, including rodents and cats. The intricate interplay between these components creates a dynamic epidemiological landscape, contributing to the persistence and spread of flea-borne rickettsiosis. The clinical manifestations of flea-borne rickettsiosis vary, ranging from mild, self-limiting febrile illnesses to severe, potentially life-threatening conditions. Common symptoms include fever, headache, and rash, which can complicate diagnosis due to their similarity to other febrile illnesses. Timely and accurate identification of the causative agent is crucial for effective management, as delayed treatment may lead to severe complications. The incidence of these diseases is often linked to environmental factors that influence the abundance and distribution of fleas and their mammalian hosts. Urban areas with high rat populations are particularly susceptible to murine typhus outbreaks, emphasizing the importance of addressing both human and environmental factors in disease control strategies. Preventing flea-borne rickettsiosis requires a multifaceted approach, including public health education, vector control measures, and improved diagnostic capabilities. Public awareness campaigns can inform individuals about the risks associated with flea bites and the importance of seeking medical attention for unexplained febrile illnesses. Vector control efforts should target both urban and rural environments, employing integrated pest management strategies to reduce flea populations. Additionally, advancements in diagnostic tools, such as molecular techniques and serological tests, can enhance the accuracy and speed of identifying Rickettsia species, enabling prompt treatment and reducing the risk of severe outcomes. Global collaboration is essential in addressing the challenges posed by flea-borne rickettsiosis. Researchers, healthcare professionals, and policymakers must work together to improve surveillance, share information on emerging threats, and develop effective strategies for prevention and control. By understanding the complex interactions within the ecological cycle of these bacteria and implementing comprehensive measures, society can mitigate the impact of flea-borne rickettsiosis and protect public health on a global scale. Please remember to subscribe to our channel, click the like button, and activate the notification bell if you wish to see more content like this. Thanks for watching.